Okay, welcome this morning to our last lab. Oh, now I'm sad. We're getting to the last of everything, and it's gonna get me all teary. Okay, so I'll try not to do that this morning. So our last lab will be on embryonic development in your purple packet. So go find your purple packet if you don't have it as you watch this. And this will be exercise 44 in the lab manual. So you can go to those diagrams. Let me find which pages those are on. Because we don't have it in the text. Wait a minute. No, because we're in the other text. So
talking about 657.
What? Yeah, 657 and 658 are the two pictures that we're going to be using, and the table down at the bottom of 658 on the derivatives of the different primary germ layers. And 660. So all of those pictures. And then you're also going to want to go back to exercise 32 because we're going to look at fetal circulation. And that is on page 447. So all of 44 and then page 447, that'll be the last thing that we do is the fetal circulation. So last semester was my embryology course. Oh my goodness, I loved that. So far, my favorites have been histology and embryology of the courses that I've taken. You know, the physiology is always really great, but that was big in all of it. I have 5 more physiology classes left, and then I will be all done taking classes academically for my life. Maybe I'll do some personal interest ones, but I'm done with school. Yeah, 50, almost 50. Well, I'll be 56 when I finish being done with school. So that's lifelong learning, friends. Always lifelong learning. Okay, so I'm going to go ahead and go to our picture. Whoops, don't do that yet. Go to our pictures and there we go. Share my screen. And well, that's lame. Okay, can you see the picture that's on 657 yes. on your screen? Okay.
Yes.
Because sometimes I don't know when I have to move things over if you guys actually get to see them or not. Okay, great. All right, let me get rid of some other stuff on my other monitor. Okay, so let's go ahead and take a look at all of these structures that are in this diagram. So next week, well, actually this week, so tomorrow we're going to start reproductive system, and then toward the end of next week, first part of May, that first week of May, we'll maybe just the Monday we'll finish off embryology. So the end of next week we'll be getting into embryology. So we'll be talking more specifically about than just identifying what these structures are. So I'm not going to take a lot of time explaining these, obviously, because we'll do that in lecture. But you know, they have to have some meaning to you. So I will do a little bit of explanation. All right, so if we take a look at this structure, right, well, I want it to be red. Let's make it purple today. All right, so this structure right here that we find right there in the first third of the uterine tubes, that is the zygote. And as you can see right there, it says it's a fertilized egg because that's what it is. Here you can see the actual fertilization process going on. So once an ovum, mature ovum is ovulated from the ovary, it'll go through the fimbriae into the infundibulum of the uterine tube. And then within that first third of the uterine tube, that's where fertilization will occur. Now, when I was young, before I started taking all these classes, I thought fertilization happened in the uterus. Well, it can't happen in the uterus because we run the risk of the zygote falling out, okay, before it has a chance to grow to the size and structure that will allow it to implant. And so you need 6 days for that to happen. So by having fertilization happen way back here in the first third of the uterine tube, now the pre-embryonic structure has, or products of conception, whatever the technical term you want to call them, they will be able to grow and get to the point where they are now able to implant in the wall of the uterus. So I think that's just amazing how, you know, everything is just so perfectly well designed to do everything that it's supposed to do. And then we have those situations where this doesn't happen. I had 2 miscarriages. Well, okay. So one was an actual miscarriage. I actually did have an embryo fetus. Now she was 12 weeks. So, but we don't think she grew past the 8-week stage. How do I know that it's a she? Well, I know that it was a she. And so there was actually a baby when I miscarried. The second one is what is interesting is when this fails to progress and you get what's known as a hydatidiform mole. And I'll show you that it secretes just tremendous amounts of HCG, which is that hormone indicator that lets you know that you're pregnant. But there is no embryo, and so I had one of those, which I didn't know at the time, because when I had my D&C, then they said, well, we don't see any products of conception, which, you know, that's how they say, well, there's no baby here. But that, how can that be, until I got into my embryology class and then learned all about it. So I'll tell you about that next week. So anyway, I digress, but let's get back to what we're talking about. So here is where fertilization happens, where the female ovum, well, the ovum from the female, is met by the sperm of the male, and then they will unite in forming this fertilized egg. So this is a single cell, and it's got 23 chromosomes from the mother and 23 chromosomes from the father. And remember too that this cell has never existed before. That unique set of 23 or 46 chromosomes actually has never existed before and will never exist again. That is a totally unique structure, and that's it. And so that's why I have a very difficult time with abortion as a means for birth control, because that was a unique individual that then failed to failed to be allowed to grow to its full genetic potential and everything that goes along with that. And so anyway, that's what that is that unique individual right there. So that's a zygote. Okay, single cell, brand new, 46 chromosomes. Now, within 72 hours, the zygote will divide a couple of times, and we'll get these blastomeres. So these are the cells that split. So this cell split twice to produce this 4-cell stage. But notice that it isn't any bigger than the original zygote, because around the outside of the zygote, we have this wall, for lack of a better term, called the zona pellucida. And so in the early stages of development, these blastomeres are going to be very tiny, because they can't go out beyond the diameter of the zona pellucida. So you'll notice here when we get to the morula, so here we are at 3 days, so about 72 hours now, we have a morula, which is a solid ball of cells. Okay, and they're tiny. They're way tinier than what the original zygote was, because they're still housed within the zona pellucida. But then we get to day 4, and we get to what's known as a blastocyst. So all of the cells in the morula are all exactly identical to one another, because no differentiation has started to happen. So remember, every cell in your body has the same 46 chromosomes, and so each and every one of those cells can be identical to one another. However, not all of the cells express all of the genes on all of the chromosomes and all of your, that you have within your nucleus. And so as cells are expressing genes more than other cells, we get what's known as differentiation. And so differentiation is when the cells take on unique characteristics because of the gene expression that's going on. And so we first start to see differentiation with a blastocyst. So what is a blastocyst? So blast is like an early form of something, like what are those osteoblasts, right? And fibroblasts and hemocytoblasts. So blast is an early form of something, not so totally differentiated. And then cyst is like a fluid-filled ball, okay? So a blastocyst are the early cells, the early embryonic cells with a fluid-filled cavity inside. So the differentiation that's taken place here in a blastocyst is that we form two layers of cells. If we take a look at this down here, this implanting blastocyst, which is essentially identical to the early blastocyst, but in the early blastocyst, you can see the zona pellucida is still there. But then if we take a look at the implanting blastocyst, it's finally hatched from that zona pellucida. So kind of like a shell, but not anything structurally similar to it whatsoever. Maybe functionally, but similar, but it's not a shell. So, and it's not even a, no. Okay, so Tony asked, is that where meiosis turns into mitosis? Nope. Meiosis is only going to happen in two places. It's going to happen in an ovary and it's going to happen in a testis. And in the ovary, when meiosis happens, it produces only ova. And when meiosis happens in a male, it only produces sperm. So this is mitosis right here. So when you went from a zygote to this 4-cell stage, this was mitosis. So this embryo will never encounter meiosis. Okay, it will never do meiosis until in males, it'll never do it until you're in puberty. In females, however, we do get some meiosis happening in the primitive ovaries, and I'll tell you about that when we get into the female reproductive system. But yeah, meiosis is only going to happen in the parent in order to produce the gametes, but it's not ever going to happen in the embryo. Okay. Because we always need, every single one of these cells right here has to be diploid. So it has to be mitosis that's carrying on this process right here. Okay, that's a great question. All right, so what are the two parts of a blastocyst that you need to know? So under blastocyst, it says trophoblast. All right, so the wall of the cyst right here, so this, I'm going to give it a different color, make it green. Okay, so what I'm tracing right here is the trophoblast. Oh, let me go grab something. Just stay right there. Don't go anywhere. Okay, I'm back. Had to go in the lab because, you know, we would be in the lab today. And that's where all my stuff is. All right. Do you guys, excuse me, do you guys remember these? Oops, you can't see. Where's the front of them? Okay, you guys know what these are? Wonder Balls? Okay, so my kids, when they were younger, so what are my kids? In their 20s. Whenever we went to Walmart, oh my gosh, these were right here at the checkout. And we would, oh, Mom, can we get a Wonder Ball? Can we get a Wonder Ball? And I'd be like, no, they're like $1.79. And that was back in the days when we were poor. And I'm like, no, I'm not buying you a Wonder Ball. So I actually have two of these. I have this one that's in my lab and I have one on my desk at home that somebody else gave me. But why do I have Wonder Balls? Because they are blastocysts. This is a perfect example of a blastocyst. So for those of you that don't know what a Wonder Ball is, it's chocolate. So can you see it's got chocolate around the outside, and then there's little candy special things. These happen to be Shopkins Wonder Balls, but there's little fun, exciting stuff on the inside, kind of like a Kinder Egg, but not, I guess. I don't know, because I've never broken open a Kinder Egg before. But so here is how a Wonder Ball is a blastocyst. So the chocolate around the outside, so all this chocolate around the outside, that is the trophoblast. Okay, so trophoblast, chocolate around the outside, the Wonder Ball, hole inside, it's hollow inside, and then the little candy things in here would be the inner cell mass. And the inner cell mass is what's going to give rise to the embryo. Okay, so trophoblast is just the wall around the outside, and part of the trophoblast is going to develop into the placenta. Now, I guess all of the trophoblast is going to develop into the placenta, but it's going to differentiate into 2 layers. So chocolate on the outside, trophoblast, placenta, and then little Shopkin candies, inner cell mass, embryo. Perfect little analogy of that thing. Okay, that blastocyst thing. Oh, I want my screen share. There we go. Okay. All right, so if we come back over to here, let me draw on this again. Okay, so here is the trophoblast. Whoops. This is all empty inside. I'm going to make it blue because it's just fluid filled. Okay, and then this, I'll make it yellow. Okay, so then this, oh, you can't even see that yellow. We'll make it purple again. This all around here. So this whole thing right here, so you can see it's an embryoblast, or we put inner cell mass. Did I put inner cell mass? I didn't even put it on your list. Okay, go ahead and add embryoblast next to trophoblast, so that you know, and then just put a little note that the trophoblast is going to turn into the placenta, the fetal portion of the placenta, and then the embryoblast, or the inner cell mass, is going to become the embryo itself. Okay, so now what? Well, now we're going to implant. And you can see in this implantation that's going on right here, that the side of the trophoblast that has the inner cell mass, that's the part that butts up against the endometrium, and it's going to stimulate the endometrium to actually surround it. So endometrium is going to grow out around that, so that eventually that embryo, that blastocyst, is totally engulfed in endometrium. To be perfectly honest, friends, yes, it is parasitism. In its best, in its truest form. So when that blastocyst implants in us, it's doing it so that it can extract nutrients from its mother, and then the process that the mother's body undergoes to engulf that uterus is to try to keep it from spreading throughout your body. So I love your kids are parasites, and they're parasites throughout their whole lives. No, they're not. I just know my oldest son, my son and his wife are moving back. Well, they're not moving to my house. They're moving to grandma's house, but they're moving back home for a year or so. So it's exciting. We're happy to have them back, but you know, they go away and they come back. So there's that. All right, let me go on to the next slide. Oh, okay, I'm not drawing with that. I have to realize what I'm drawing with here. Okay, so oh well, let's just, okay, so there's the zygote stage, excuse me, 4-cell stage. There's the morula, there's the early blastocyst and the implanting blastocyst. Okay, so now we're going to take a look at what happens once implantation has happened. So we're going to get some cool stuff going on here. So the first thing, I think I'll draw with this one. Oops. Nope. Okay. Oh, it's not. All right, never mind. I'll draw the other one because it's not letting me. All right, so here is your trophoblast. right here. Now look at what's happened to it. It's not just cellular. Well, it's not just single cellular anymore. It's almost like a tissue. And what happens is that the cells, let me just draw a trophoblast back down here again. And we'll talk more about this next week. Ugh. Okay, so here are all the cells, boom, with nuclei in them. Okay, so what these cells are going to do is divide, and then as they divide, so here would be this cell dividing, this cell dividing, we lose the membrane between them. So they become big clusters of many nuclei. So you can see here all the nuclei, but there's no cell membranes between them. It's just one big mass. We call it a syncytial trophoblast. So what that means is that the cells have come together, syncytial trophoblast. So all the cells of the outer layer of the trophoblast have come together to form the membranes here that separate maternal blood from fetal blood. Okay, so they make almost like a blood uterine barrier. Well, I guess that is. It's a blood uterine barrier, basically. All right, because we never want maternal blood and fetal blood to mix during fetal development, because antibodies from the mom are going to come and attack the baby. So we don't ever want that to happen. Okay, then the trophoblast is also going to become a second layer here that's called the cytotrophoblast. So this cytotrophoblast here is this red layer, oops, that goes all the way around here. So here is that same cytotrophoblast. And if you look at this right here, it says chorion being formed. So I have these little buds. Can you see how there's little buds that are coming off of this cytotrophoblast right here? Okay, the chorion is the vascular layer of the fetal placenta. So all of the fetal blood that's going to, or all the blood that's going to be in the fetal side of the placenta that's going to exchange with the mom, is nutrients and waste products with the mom, is going to come from the chorion. And the chorion comes from the cytotrophoblast. So here we can see the chorion. So all of this red layer right here is all chorion. Okay, so it's the second layer of the trophoblast, and then we get this extra embryonic. Well, coelom is a cavity, but we get this extra embryonic mesoderm out here, and then we get, see that right there, and then that gets complicated, but we're not going to worry about that for you guys. So there's the chorion. We still have a lot of stuff in here, and the baby is going to be this right here, which we'll get to in just a second. Now the chorion is going to develop these extensions called chorionic villi, or one is a chorionic villus. And what these chorionic villi are going to do then, if we take a look at 4.5 weeks right here, look at what all the villi have become. And actually the process of this villus development is really kind of cool too. Obviously, embryonically, we talked about that. So, because we have primary, secondary, tertiary villi, and that these are tertiary villi, ones that are actually developed enough to the point of capillary beds in these villi where exchange can happen. And then this from the outside, this is the maternal blood. So these are spiral arteries within the uterus, within the functionalis layer, that are then going to come down and form this these sinuses right here. So they're big places where blood pools, and then exchange of gases will go back and forth across there. And we'll talk more about that next week. All right, now at this stage, so if we go back to 7.5 days, let me get a different color. Pink. Okay, you were a 2-layered embryonic disc. You had an epiblast and a hypoblast. The epiblast was blue and the hypoblast was yellow. And then, so this came from the let's make the inner cell mass. Okay, so here is my inner cell mass right here. And it's a whole bunch of cells. Okay, so what I'm going to do is I'm going to start to get a little cavity inside of here on the top side, and then I'm going to get a cavity down here on the bottom side within these cells. this is going to develop into the amniotic cavity. Amniotic cavity. All right, so there's going to be fluid all in here, and the cells that are going to be up here are all the amnion. Where's my amnion? There we go. So these blue cells right here are the amnion. And then down here, we're going to make a yolk sac.
Miss G, you need to buy one of these Oreo eggs that came out this year. Look at this thing. Ready?
Yeah, just a sec. I got to stop sharing. Okay.
Okay, here's your Oreo sac, or
Okay.
your Tropoblast.
My trophoblast.
Now look.
Oh, yay.
There you go.
Very
And
good. That works too.
have Oreos inside.
Excellent. All right. So anybody come up with other examples of blastocysts? I'll be very happy. I was going to say give you extra credit, but no, I won't. Okay. So thank you for sharing that. That's awesome. All right. Oh, I just lost all my drawings. Oh, well, we'll draw some of them
I'm
back.
sorry.
No, you're fine. Okay, so let me get my trophoblast here, and then here was my inner cell mass right here. This will be fine. So we had this cavity on the top. That was the amniotic cavity. I'm just going to put A for amniotic cavity. And then down here was the yolk sac. And if we were chickens, then we would feed off of that. Okay, there's the yolk sac. But we're not. We're placental animals. So we're going to develop a placenta, which we'll get to in a minute, and see how we're different. But then the cells that are here, so this kind of comes, so this will go all the way out to here, actually. And this will go all the way out to here. So then the cells that are stuck in between the two are going to develop into two layers. So we're going to have the epiblast is all up here, and then the hypoblast is all down here. So you're this two-layered embryonic disc right there, okay? And then you become a three-layered embryonic disc. So the epiblast turns into ectoderm, and then it's going to actually also differentiate into mesoderm, and then the hypoblast is going to be endoderm. Okay, so we get three germ layers that are then going to give rise to all the rest of the cells in the body. But associated with, so to make sure we got all of our, so we got amnion, so there's our amnion or amniotic cavity, and the membrane right here, it should be blue, but anyway, there's the amnion. So when you get your membranes, you know, when your water breaks and your amniotic membrane breaks, that's what's breaking, is this blue thing right here to let your amniotic fluid out. All right, and then the yolk sac is down here. So we've got lots of other functions for the yolk sac, where our gonadal tissue and our, what else comes from the yolk sac? The very first blood in the baby comes from the yolk sac, but we'll talk more about that. And then we have the allantois. And the allantois, I'm going to show you more in just a little bit, but here it is. That's going to come up here into what's going to become the umbilical cord, because we are going to have the part of the allantois is going to be in the umbilical cord, and it's actually going to be part of the bladder of the baby, and kind of like holds the bladder in place. And there's sometimes this turns into a structure called the urachus, and if the urachus persists and stays open in the umbilical cord, sometimes newborn babies will leak urine out through the stump of where their umbilical cord is, because the urachus didn't entirely close off. So that's kind of crazy, but that does happen. So anyway, okay.
Does that end up healing on it and like closing on its own, or is there a surgery that would
They'll probably just go in and stitch it up to make sure that it's all closed up. Yeah. Okay, so let's, oh, I have to get rid of that. Okay, so let's move on. this is a nice picture. It shows how the maternal blood here will then diffuse, the nutrients and waste products will diffuse back and forth across the fetal capillaries in the chorionic villi. You can see how they're really bathed in maternal blood. So there's actually these big sinuses open in the decidua basalis that allow that exchange to take place. All right, so if we look down here, we can see umbilical arteries, umbilical vein, connection to the yolk sac. There is the allantois right there. And well, at this stage, it would be a urachus, but anyway. So we're gonna take a look at fetal circulation now. So we're gonna look at what these blood vessels are doing, their jobs. Okay, now one thing I want you to notice. So what do arteries always do? Carry blood away from the heart. So if we look at the arrows here in the umbilical arteries, we can see, oh yeah, they are going away from the embryo, but, or not an embryo, fetus at this point. But look at what color they are. Usually arteries are red because they contain oxygenated blood. These, however, do not contain, they are fairly deoxygenated. Because remember, when you're inside your mom, you are not going to be doing gas exchange in your lungs. You'll be doing gas exchange right here in the placenta. And so this is the blood that's taken, the fetal tissue has taken the oxygen out, and so now we're going to get it exchanged and come back. So now as the blood is returning back to the embryo, fetus, sorry, you can see, okay, coming back, but it's now red because it has been oxygenated, because this is where gas exchange happens for a fetus, is in the placenta. Okay, so this whole thing right here is an umbilical cord. Let me get my little writing stuff. Okay, so umbilical cord right there is this entire thing. And you need to know that in an umbilical cord, there are 2 umbilical arteries, yet only 1 umbilical vein. Okay, and then this little, what's left of the allantois and the urachus right there. Okay, so let's now go to 477, and we want to look at fetal circulation, because that's really all we got left in here. Okay, so let me see what's next here. Okay, let's do this now. Let's talk about these things before we go into fetal circulation. So the 3 germ layers and their derivatives. Okay, so it's hard to say specifically, well, I guess it's not. You can, okay, yeah, you can, because like with the kidney, where's kidney? Okay, kidneys and ureters develop from mesoderm, but the bladder, where is it? It's not on here. Urinary tract, there we go. So bladder and urethra come from endoderm. And what happens is that they plug into one another. So the ureters will plug into the bladder of the urinary tract. So the kidneys and ureters will actually come from mesoderm, but the bladder and the urethra will come from endoderm. So some organs are made from 2 different kinds of structures. Okay, so and again, we'll talk more about this in lab, but you might as well start learning these. So I would start memorizing what comes from what from this chart. Now I won't go any more detailed than this chart in lecture, on the lecture exam. Okay, so ectoderm. So ectoderm is the outermost layer of the embryo. Okay, of the 3-layered, the tri-layered embryo. Embryonic disc is what we call it. So the outermost layer, the ectoderm, remember we already saw the nervous system. So think back when we were doing 201, and then coming down on your back, remember that your back folded in, and then the tube dropped off, broke off, and became the neural tube, which developed into the central nervous system. And then also special senses. So your eyes grow off of that. Part of your hearing structures are going to come from that. And then, so if this was on your back, on the dorsal surface, okay, so it came, so epidermis, because that's on your back too, is also going to come from ectoderm. So just the epidermis and then this accessory skin structures, those are all the epidermal derivatives. So hair and glandular, so sweat glands and sebaceous glands, those are all going to come from ectoderm. Now, think about outside all the time now. So outside, so the epithelia of your oral cavity, I'm still on the outside. Remember, I come from skin, I get into my mouth, and then I just curve around my lips, and I go into my mouth, down into my esophagus. Well, I start to get into my esophagus, so down into the pharynx. Nasal cavity as well, and anal canal, because I'm going to come from the bottom, and then I'm going to curve up into the anus, and then line the anus as well. Now, these these three things, the adrenal medulla, the pituitary, and the pineal glands, particularly the adrenal medulla. So these are going to come off, obviously, off of the brain, the CNS. Okay, but this is kind of cool. So we have on the sides of our spinal cord, we have these, or this neural tube, actually, we have these clusters of cells called neural crest cells. And neural crest cells actually give rise to a lot of different things. But what they do is they leave the area of the back, and then they travel down into the adrenal gland and form the adrenal medulla. So all the epinephrine, the chromaffin cells that produce epinephrine and norepinephrine that are, you know, so important in sympathetic stimulation, then are going to come from the adrenal medulla, but there's still a connection there. So as the neural crest cells come from your back, from that neural tube, and then come down, now they're plugged into your central nervous system and into your medulla, or your adrenal medulla. So that's how we activate sympathetic stimulation from our spinal cord and then down into the adrenal medulla, and then we start kicking out epinephrine and norepinephrine. Okay, the mesoderm here, let me get a mesoderm color. Okay, so the mesoderm, I'm sure I'm gonna go back and forth between mesoderm and mesoderm, because my embryology professor kind of did the same thing. He mostly called it mesoderm, but they would call it mesoderm sometimes. So it's the same thing. Just what I don't know. Anyway, so I'll just switch back and forth. Okay, how do I remember mesoderm? Muscle and bone. And middle. So middle muscle bone is what gives rise, or what mesoderm gives rise to. Okay, so middle, oh yeah, look at that. There's my dermis. Okay, so it's under my epidermis. It's in the middle. Gives rise to my skeleton. Gives rise to my muscles. Okay, gives rise to cartilage and my blood vessels. Oh, and then mesoderm. What word do we remember? Mesenchyme. Mesenchyme comes from mesoderm. See, there I switch back because they go together. Okay, kidneys and ureters, intermediate mesoderm. Lymphoid organs and tissues, internal reproductive organs. These are going to be the gonads. So the ovaries and testes. And actually these guys and these guys come from the same stuff, our serous membranes, and adrenal cortex. Okay, so it's going to be in the middle, and I mean in the outside part, but this is going to get poked in by ectoderm. So this would form a cortex on the outside like this, and then the neural crest cells are going to poke down into the center of it and make a medulla. And there's the cortex. Okay. Now, endoderm is on the inside. Okay, so endoderm is what's on the inside of you. Well, the lining of, so remember, your respiratory, well, let's do GI tract, because we know the layers of our GI tract. So we have mucosa, submucosa, muscularis, serosa. So the serosa of the GI tract and the smooth muscle of the GI tract is all going to come from mesoderm. It's going to come and surround my original tube that was the epithelium So this would be the simple cuboidal, no, columnar. Okay, so the simple columnar epithelium of the GI tract is going to come from endoderm. And really, let me draw you this picture. So sorry, you guys, if it wasn't for the fact that I just took this embryology class, I probably wouldn't be going into this detail, but I love it so much. And you should love it too. All right. So I'm going to draw what you are. Green is good because I'm not going to use green. Okay, so here you are as an embryo. You look just like that. Aren't you beautiful? And you started out, well, actually started out down here because you're a deuterostome. You started out with a gut tube. And you had a gut tube. Actually, I should have made that blue, not this blue. Let me do this blue because this should actually be blue. There we go. Okay. So this should be blue. So here you were, your two layers. This was your epiblast and your epiblast and your hypoblast. Okay. And so this was you. You were just these two layers stuck together with a hole down the middle. This was your gut. And then in your gut, you had a whole bunch of things. So you had your epithelium, so your simple columnar epithelium, but it's going to differentiate into a lot of other tissues. So really, they were more like blasts than anything else. Okay, so then what does it say comes from endoderm? Respiratory tract. So here's your mouth. Here's your anus. And so off of your pharynx, so to about here would be your pharynx. So then what happens? Oh, I grow some lungs off of that. Hey, look, I do. Okay, and then this is going to curve and it's going to become a stomach and then intestines and everything. So we get down here, which is going to become a cloaca, which will then split and you'll end up getting a little, so we put a little split in here, a urorectal septum, and you'll get a bladder that comes off of that. So urinary tract, bladder, and urethra. So we get like I said, we get a wall right in here. Reproductive tract, if you're a girl, then we're going to get a vagina back here too. And then liver. So we're going to sprout a liver off of here. We're going to sprout a gallbladder off of here. We're going to split a pancreas off of here. And we're going to split a thymus off of here. That'll grow right here. And we're going to get a thyroid gland. It's going to come off up here, and some parathyroid glands. And so, and then up here, it's beautiful. You're so lovely. So here is going to be your brain and your spinal cord. And then in here, we're going to get these somites that are going to form all the way along your back. You get like 33 somites. And then they're going to grow out and make all of your muscle and all your bone. Because you have a muscle component and a skin component and a bone component to each somite. Dermatome, sclerotome, myotome. And those will all develop into the bones and muscles and underneath the dermis of your skin. So that's what you've developed from. Oh, it's so fun to learn how all that happens. So anyway, that's how all that happens. Okay, so now we're going to go to page 477. So I need to find that of me. Oh, wait. Oh, that's just heart. I gotta go somewhere else to find my thing. Oh no, here it's still. Oh, okay. 32. I'm waiting for something to download. Okay. Extract all, extract. Okay, and then view large. Okay, here we go. All right, so let's look at my screen. All right, so here we are with a comparison between fetal circulation and newborn circulation. So here's the fetal, right there. There is the newborn. So the first thing that you notice that's present in fetal circulation that is absent in newborn circulation is the placenta. So here is the placenta there, excuse me, with the umbilical arteries and veins and their fetal divisions here on the fetal side. So all of this vascularity right here, this is all on the fetal side of the placenta. If you look at the maternal side of a placenta, it looks very different because it's not as vascular as all of this is. Okay, because this is, think about, this has got to go down to all the capillaries that you find, that you would find in an adult or in a newborn, in the digestive tract and in the lungs, because that's where all the exchanges and the absorption are going to take place. Oh, and the kidneys too. So yeah, actually the fetal kidneys do some urine production because fetal kidneys produce most of them in the embryonic fluid. Amniotic fluid, sorry. And so babies are drinking and peeing all the time. And we know that if you have some oligohydramnios, which is not enough amniotic fluid, it's usually going to be because there's a problem with either with the kidneys or there's a blockage in the formation of the trachea and the esophagus, so that can't circulate through the baby. Okay. Anyway, I digress. Okay, so here's my placenta, here's my umbilical cord. So you need to know what 3 things are in the umbilical cord. Okay, so there are two umbilical arteries. So that's these pale pink things right here. So pale pink in this picture means low oxygenation instead of very low oxygenation, because there's still some oxygen in here. It's in this inferior vena cava and superior vena cava of the fetus that will be very low in oxygen. Okay, so 2 umbilical arteries, 1 umbilical vein, big fat red thing. Okay, so here it is, big, large, 1 large umbilical vein. Okay, now we need to get to the baby from the umbilical vein because we're not normally plumbed for that, because obviously you can see, yeah, I don't have a vein that's going from my umbilicus up into my inferior vena cava. So we need a connection. So here's our umbilical vein right until here. So it's going to stop right here at the liver, and it's going to branch into these hepatic veins right here. But it's going to become, so all of this, so from here to here, all of this part right here is called the ductus venosus. Okay, so the ductus venosus carries blood from the umbilical vein into the inferior vena cava, and it's going to pass underneath the liver. So we're going to have some branches that go off to take fetal blood to the liver, obviously, but we need to take most of that blood out here to the inferior vena cava. Okay, so ductus venosus. And how are we going to remember? It's a duct that goes between veins. So it's going to be between the umbilical vein and the inferior vena cava, ductus venosus. All right, so now that blood that's just come from the placenta and it's just received all of its, nutrients and oxygen, everything is going to mix with the blood coming from the inferior vena cava and some of the blood coming down from the superior vena cava, and we're going to go into the right atrium. Okay, so just like always, the blood's going to go into the right atrium. Now, because the lungs are ineffective for gas exchange, we want to bypass the pulmonary circulation, and we want to proceed on into systemic circulation. So there's an opening right here between in the interatrial septum between the left atrium, or the right atrium and the left atrium, called the foramen ovale. Now I'm going to go ahead and stop sharing that for a minute because that's so tiny, but I want to show you a better picture, of the foramen ovale. And it's actually two membranes. So we have two membranes that come from that form the walls of the atrium. Because remember, an atrium, we have a 4-chambered atrium, but embryonically it's a single chambered atrium up at the top. We have one atrium up the top, we have one ventricle at the bottom, and then during embryonic development we form the two septa. What am I trying to do here? Okay, so let's go to these images. These are good, and we'll go ahead and share that. That would be Aaron. Hi, I'm recording. What can I do for you? What? Okay, well, when I'm done. Okay, bye. He wants a soda. Okay, so here is how your foramen ovale works. I want this picture. Oh, maybe we'll get it here. There it is. Can I get that bigger? I'll just make it bigger this way. And then we'll scroll down. Okay, so can you see that? Am I sharing that? Not now. I am now. Can you see this? The foramen ovale? Okay. All right. So here's what's going to happen. So I have blood. Oops. I can't draw on it. I have blood. Maybe I can now. Yeah. That's going to go over here. I do have some that goes this way, but a vast majority of the blood is going to go over here from the right atrium into the left atrium. And then I can hurry and send it out into systemic circulation. So it says a small hole located in the septum, the wall between the two upper chambers, as you can see there. And then it says, before a baby is born, it does not use its lungs. This is not written for you guys. This is written for normal, everyday people. You guys are above that. It does not use its lungs to get blood rich in oxygen. Instead, this blood comes in cave. So blah, blah, blah. The foramen ovale makes it possible for the blood to go from the veins to the right side of the fetus's heart, then directly to the left side. And then the foramen ovale normally closes as blood pressure rises in the left side of the heart after birth. And it does. It just like pretty much snaps shut. This particular website, though, is about what happens when it doesn't. It's a patent foramen ovale. And we'll talk about patent foramen ovales later in lecture. Okay, so
Hey,
that,
Misty,
yes.
so do they not use their ventricles either
Yes, they do.
until they're born?
Nope,
Okay.
we're gonna talk about that right now, but that's a great question,
Okay.
Joanna. So let me just go ahead and incorporate that into where the blood's gonna go. So you would think, well, if it just goes over the other side, then that's okay. But no, it doesn't just do that. So here is what's going to happen. We'll do it in orange, and then we'll magnify. No, it won't let me do it with my thingy. Hang on. Okay, so magnify, boom, and then we'll pull it down. All right, there, that's a much better way to look at the heart. So here's what's going to happen. So the blood's going to come down here and up here, And then a big bunch of it's going to go over here. I don't know, 60, 70, 80%, something like that. But notice too that it comes down here. Okay. So you can see it does go down into the right ventricle, and then it is going to go out the pulmonary trunk to the lungs, because the lungs themselves, they need blood. And even though the lungs are not going to get, the lungs actually get blood through the bronchial arteries that come from the thoracic aorta. But we need to get this whole circulation plan going. So we're going to practice. I mean, it's basically just practice. Send the blood out, bring the blood back. So it's going to go down into the right ventricle. It's going to go out the pulmonary trunk. But here is our second bypass called the ductus arteriosus. I guess we have 3 bypasses. So we have the ductus venosus, connect the umbilical vein with the inferior vena cava right there. And we have the foramen ovale, so the bypass between the right and left atria. And now the ductus arteriosus. So then the rest, almost all of the rest of the blood, so I don't know, maybe a 30% or something like that, instead of then going out to the lungs, is then going to go right into the arch of the aorta. So there's a connection between the pulmonary trunk right here, which is an artery, and the aorta, so that's why we call it the ductus arteriosus. So once the blood has been out here, then it's going to come back and go in here to the left ventricle, or it's going to pass over here down into the left ventricle and then go out through systemic circulation. So yeah, we do use the left ventricle to pump all of the blood out, but we only use the right ventricle. Well, we pump all the blood out through the right ventricle, but instead of it going all to the lungs, because remember we told you it goes all to the lungs, it's going to go to the left atrium instead. Okay.
When does the foramen ovale finally close?
At birth.
At birth it closes.
Yes. So when you take in that first breath of air and you develop more pressure in the what are these? Pulmonary veins on the left side of the heart, that slams this shut and it sticks shut. And so it pushes back this way, which closes up because the holes are not offset. There's a hole at the bottom and a hole at the top, and then membrane. So you know, in that other picture, it showed that curvy arrow. So the pathway is S-shaped. So as soon as you increase the pressure over here on the left side, when you take that first breath of air, then it slams them shut, and then they stick shut. Sometimes,
is that like the part that babies are having like surgeries on because those aren't closing?
No, when babies are having surgery, it's because the ventricle or the interventricular septum didn't close. Because if you notice right here, and usually that hole is right there, because I have a membranous part of the interventricular septum, and then I have a muscular part.
Mm-hmm.
So let me clear all of this mess. Okay, so this comes down to here, and then this comes up to there, and they fuse. Sometimes they don't. And so when babies have holes in their hearts, then we go from ventricle to ventricle, and that's bad.
Okay.
Now we're not pumping.
It's not going anywhere really.
Out to the lungs, right? It's just going over here to this other side. Sometimes if your patent foramen, or if your foramen ovale doesn't close like it's supposed to, then your baby can be purple, and Cameron was purple, and I'll do that in lab. I mean, I'll do that in lecture. So we'll talk about that in Cameron's case. And
then the ductus arteriosus and the ductus venous, do they just disappear?
So the ductus arteriosus closes pretty soon after birth too, and the ductus venosus takes a couple days, and then it closes off.
Interesting.
Yeah, and we'll go into those closing off structures right now, I guess. Okay, so does anybody have any questions now about up to this point? Any other questions? Okay, so we're going to take this back down, and now we're going to look at the leftover things. So what do we call the ductus venosus after it closes up? Okay, so this is going to be over here in oops, I went too far with that arrow. Over here in the newborn. So here is my ligamentum venosum, right there. My umbilical artery actually becomes the ligamentum teres, but I didn't make you know that. Okay, and that's going to be hooked into your umbilicus. And then we have the umbilical arteries become median umbilical ligaments right there that come off of the common iliac artery there. And then ductus arteriosus becomes the ligamentum arteriosum, which sounds very much like a Harry Potter spell. In the heart model, there was a little white line, little white connection between the pulmonary trunk and the aorta. So that was the ligamentum arteriosum. And then it'll just become a little dip right here called the fossa ovalis. Because remember, a fossa is a depression, but a foramen is a hole. Okay, so now it's just that oval depression there in the wall between the left atrium or the right atrium and the left atrium. But like I've kind of mentioned here and there, is that it can stick open, and then you have a patent foramen ovale. But again, I'll talk about that in lecture. Okay, so that's that. That's our list. Nothing else. So, oh gosh. Okay. Did we? We didn't, did we? We didn't do Physio X9. We did Physio X7 and Physio X8. I know we've been over those. Physio, oh, there's that I was looking for. It's in my book. Oh no, we did, yes, because that's why my nail file's in here, because I said speculate on ways that desert rats are able to concentrate their urine significantly more than humans. Didn't we go over 9?
Yes, ma'am.
Yes, ma'am. Okay, that's what I thought. Okay, so 10. Oh yeah, because I want, okay, yeah, we did all this. So Physio X10 is just acid bases. This is all easy. So go ahead and go to, we'll just look at ourselves while we do this, because I don't have a thing. All right. Okay, so on page 1, PEX 151 is that the first question? Yeah, so this is hyperventilation. So the first activity, so in the activity questions, it says, at what pH range is the body considered to be in a state of respiratory alkalosis? And I'm just going to go through these really quick. So greater than 7, nope. Less than 7.3, nope. Less than pH range, yeah. Oh my gosh. Read the question. At what pH range are we in alkalosis? Greater than 7.45, which is what I said in the first place. And then I was thinking about hyperventilation. Okay, how can the body compensate for respiratory alkalosis? We're going to hang on to hydrogen ions, or we're going to get rid of bicarbonate ions, right? Because we need more hydrogens. And then we don't care about, how did the tidal volume, well, actually, yeah, we can do that. Number 3, how did the tidal volume change with hyperventilation? We breathe faster, but we also breathe deeper. Okay, and then what might cause a person to hyperventilate? Getting ready for the test this week. Sorry. I don't know, COVID-19, really, and that was being rude, but yeah, I mean, I'm sure people that are having those lung issues are trying to get that oxygen, they just can't. So I'm sure that our serious COVID-19 patients are in respiratory acidosis and trying to get rid of that, so they'll be hyperventilating to try to get the oxygen to them as well. Okay, then on the next page, what will happen to pH and PCO2 levels during rebreathing? So this is when we're going to breathe in a paper bag. So think about, why would we breathe in a paper bag? We're hyperventilating and we're blowing off all of our CO2. So now there's no stimulus in the dorsal respiratory group for breathing, because I just blew off all that CO2. So I have to build that back up again. So that's why we breathe into a paper bag, to stimulate respiration. And so if we're breathing in the paper bag or rebreathing, our PCO2 levels will go up and our pH will go down. So always remember, high PCO2, low pH. Let's see. So did the pH change with rebreathing? Yes, it goes down. What happens to pH when there's too much carbon dioxide in the blood? It goes down. Tidal volumes change with rebreathing. They're going to go more back to normal. And then 2 ways in which too much carbon dioxide might remain in the blood. So COPD or, emphysema, COPD, so chronic bronchitis. Okay, so those are that. Okay, then on 153, this is renal response to respiratory acidosis and alkalosis. So what effect do you think lowering PCO2 will have on hydrogen ions and bicarbonate in the urine? So if I lower PCO2, I'll have less hydrogen ions. We're not talking about pH. I have fewer hydrogen ions and more bicarbonate in my urine. And then raising CO2 will have more hydrogen ions in my urine because I'm trying to secrete those because I'm retaining CO2, and then less bicarbonate. How the kidneys respond to respiratory acidosis: secrete hydrogen ions, retain bicarbs, make new bicarbs. What PCO2 corresponds to respiratory acidosis greater than 45? Describe how the kidneys respond to respiratory alkalosis. They retain hydrogen ions, they secrete bicarbs. And what PCO2 corresponds to respiratory alkalosis less than 35 millimeters of mercury. All right, and last but not least is metabolic acidosis and alkalosis. And so this is actually looking at metabolic rates. So it's not looking at I'm starving or I have DKA or I'm an alcoholic or something. This is just what is our metabolic rate. So I'm exercising, I'm not exercising, I'm sleeping, something like that. Okay, so on page 155, what do you think will happen when the metabolic rate is increased to 80 kilocalories per, I don't know, per hour? So I'm exercising. So what's going to happen to my metabolic rate? Am I going to make more CO2? Yes. Okay. So then what do you think will happen when the metabolic rate is decreased to 20? That's when I'm asleep. I'm not doing anything. So it's going to, what does it say? Happen when the metabolic rate. Oh, so metabolic rate is high, my pH is going to drop. And when my metabolic, when my metabolic rate is low, my pH will rise. Okay. So, in question 1 on the last page on 156, describe what happens to carbon dioxide and pH with increased metabolism. CO2 goes up, pH goes down. Describe the respiratory response to metabolic acidosis. So think about it. You're exercising. What are you doing after you exercise? I was doing a more anaerobic activity exercise workout this morning. Wasn't very cardio at all. And I was breathing crazy heavy. And I thought, holy crap, I'm so out of shape. And I've been working out twice a day since the whole COVID-19. So I don't know what's happening, but I was breathing really heavy. So why was I? Because it was such a heavy duty metabolism generating kind of activity. I was making lots of CO2 and my pH was dropping. Okay. So then I had to hyperventilate to fix the problem. I had to blow off CO2 and raise my pH. So that would be it. Hyperventilation is a response to to metabolic acidosis. And then when the respiratory system compensates for metabolic acidosis, does the pH increase or decrease in value? It's going to increase. Okay, so when I am in metabolic acidosis, respiration will act to raise pH by blowing off CO2. And then describe the respiratory response to metabolic alkalosis. So think about, I got high pH. Do I want to blow off CO2? I want to retain them. So think about when someone's asleep, not when they're snoring. That's a whole different story. When there's super deep sleep, super low metabolism going on, can you even tell that they're alive? Sometimes not. Okay. So my little 94-year-old mother, oftentimes I go in when I drop the dog off in the morning, I'll go down into her bedroom and I'll look to see, is she still alive? Yes. And so I'll have to wait because she's breathing so slowly and so shallowly that I'm like, is she okay? And then I see the blankets move or the dog jumps on the bed and then she stirs and then I'm like, okay, because I don't want to wake her up. I don't want to say, hey mom, are you still alive? But that's what I'm doing, and that's why. So your metabolism is so low that you want to retain CO2 and you want to lower the pH. Boom, we're done with lab, friends. Yay, are we happy? Look at Nicole's little face. No, I know. I'm sad. Okay, so here's what's going to happen. We will have questions on the lab practical from the PhysioX, okay, that we've gone over. So review those questions. And since we haven't been able to cross terms off very adequately, because like I said, people haven't taken their quizzes like they're supposed to, I will make you a list and I'll post the list so that you will know, okay, I don't need to study these terms, you know, without giving too much away to people who need to get on the ball. I'm not mincing words anymore. When have I ever minced words? Like never. Okay. So does anybody have any questions then about the lab practical? It's going to be online too. It's going to be 50 questions, just like your quizzes, just fill in the blank, and that'll take me a little bit longer to regrade. So please bear with me.
How long will we have to take that?
You have until the 28th to take that.
No, like how much is the time?
You'll have
frame?
an
Is
hour.
it 2 hours?
You'll have an hour. Okay. I'll give you 90 minutes. Is that good? I'll give you 90 minutes. I'll make a note right now because usually I just give people an hour, but just for you, Nicole.
Thank you.
90 minutes. Lab practical, lab 2. Okay. And yes, Ella, you're very welcome. I'm thinking of you guys mostly. But you're not the only ones that like to stay. So that's okay. Okay, let's see. So there's that. Anything else that you have specifically about the lab practical? Okay, so are we still recording? Yes, I just have to make sure. I'm so paranoid that I'm not going to remember because Macy's not here. Nope, Macy's not
Yes,
here.
she is. Macy Bingham.
Yeah.
She's here.
You're not, oh, there you are. You're up next to me. I'm looking for your M and I was looking down below. I wasn't even looking next to me. Okay. So our next question from Kaden was, when does the lab practical open? Tomorrow. 22nd. Yes. 21st. Well, maybe today. Maybe this afternoon.
So the test and the lab practical will open tomorrow?
The lab practical might open this afternoon. I have some questions that I need to change. There is one that's ready, but after I finished it yesterday, I was like, no. So it'll be open tomorrow. We'll just straight up tomorrow at 12:01 a.m. It'll be open until the 28th. And then your test is open through 11:59 on the 25th, your test number 5. And there are 5 ABG questions on it. So you can get 5 extra credit points on that by remembering your causes stuff. So, okay. So any other questions before we go Kahoot? Okay. Then, where's my Kahoot? Right here. Oh, let me share. Okay, where is it? I just saw it, you guys. Oh, there it is. Okay, play, teach, classic. There you go. Okay. So 199-6583. What haven't we played in a while? Trance. What's beatbox sound like? Oh, I kind of like that. How's that different from reggae? No, it's less intense, I guess. Oh, it's funk. Oh, yeah, that is funk. What's trance? Sorry, I'm just playing while you guys are getting in here. Oh, that's like Sandstorm. I don't want that. Oh, I work out to that kind of music. I like the beatbox one. Okay. Oh, that's pretty.
You work out to really slow music?
No, not this. This is
I thought that's what you were saying. I'm like, that's horrible.
This is what I work out to. I have a Sandstorm radio. See, this is probably 100 and I don't know. It's over 100. But okay. There we go. We'll listen to some funk. All right. I don't know if I'm gonna get anybody else in here. Let me know if I need to wait and then any more.
More people need to jump in, because if not, I'm going to be 12 out of 12 here.
All right. Well, that's gonna be it. Then I'm gonna go ahead and get started. All right. Here we go. Okay, blood supplied to the nephrons through a structure called. Oh, I mean, it's the answer's there, but it's just not subject verb agreement. Should be glomerulus because I put a structure, but yeah. So blood's in the glomerulus. There's no blood in the capsule. There's no blood in the tubule, and so you don't supply blood through a nephron. Okay, so glomeruli or glomerulus, single one. All right. Urine forming units of the kidney are yes, it is the nephron. Okay, so we have to have everything because the nephron is the collection of the capsule and the glomerulus and the tubules and the loop of Henle and everything. So nephron is the answer for the unit of the kidney that's going to do that. Okay. All right, the vasa recta are associated with which structures in the kidney? So where do we find the vasa recta? Yay, good job, you guys. It is the juxtamedullary nephrons. Excellent. Okay, juxtaglomerular cells secrete something while the macula densa release something else. Good, not a good job. You guys, where does aldosterone come from? The adrenal cortex. Silly. Okay, so yes, juxtaglomerular cells release renin, but remember the macula densa cells are going to release a vasoconstrictor when there's too much sodium chloride and the glomerular filtration rate is too high. So then they'll secrete a vasoconstrictor to shut that down, slow that down. Okay, so no. Aldosterone is only made by the adrenal cortex. Why is there a higher filtration rate in the glomeruli when compared to other capillaries? Perfect. That is exactly right. we have fenestrated glomeruli, so more stuff comes out. We've got lots of holes, those filtration slits in the visceral layer of the glomerular capsule. And yeah, the efferent arteriole is smaller than the afferent arteriole. So blood sticks around longer to create greater hydrostatic pressure. Nice. All right, which of the following substances would not be found in normal filtrate? Filtrate. Not urine. Proteins. Yep, we should find everything else in our filtrate, right? Because everything else filtered out, but we should never have protein in our filtrate. All right, what would the afferent arteriole do if blood pressure increased? So if the pressure coming into the afferent arteriole got to be too high, what would it do? Good. It would vasoconstrict, myogenic mechanism. All right. Autoregulatory mechanisms are most effective. So when would our kidney want to just take care of the problem itself, so that we're not going to mess with all the other systems of our body? Okay, so this one, if our arterial blood pressure dropped below 80, when we have the blood pressure cuff on us, sympathetic stimulation is going to kick in, and that's going to be systemic, right? So sympathetic will affect my whole body. But if my arterial blood pressure is in a somewhat normal range for the rest of my body, then I'm going to use my autoregulatory mechanisms to adjust nephron by nephron. Okay, so when my body is in a somewhat normal range, I'm going to use autoregulation. But when my blood pressure is below a normal regulation, a normal level, then I'm going to kick in systemic activity, so sympathetic stimulation. All right. Okay, extrinsic controls regulate glomerular filtration rate as a means of regulating. Okay, so extrinsic controls. Autoregulation was intrinsic. So what do the extrinsic controls control? Yes, they go out in the rest of the body. Perfect. Ooh, TMA, way to go. Which of the following results from the renin-angiotensin system? The first two. I guess I meant red and blue. Yes, red and blue. Good, because that's what, and so yeah, you guys were right over here, blood pressure elevation. And how do we raise the blood pressure? By conserving water. And how do we conserve water? By raising blood sodium levels. So that's why that one's not part of the answer. Okay, why is glucose in the urine an indicator of diabetes mellitus? Ooh, okay, nope. All right, why is glucose an indicator? Because the high blood glucose levels would exceed the transport maximum of the nephrons. And so we drew it, it was the, Drake and Josh in the sushi factory, or Ethel and Lucy in the chocolate factory. Okay, so too much glucose was present. We can only extract so much. The rest is going to end up in our urine. Okay, so remember this. When you're diabetic, your glucose levels exceed the transport maximum. You can't reabsorb it all. Okay. All right. Two main fluid compartments within the body are what and what? Yes, intracellular and extracellular. This one, plasma interstitial fluid, that is just extracellular. We have to remember that 62% of our body fluid is intracellular within our cells. All right, which route accounts for the most water loss, second only to urine output? Oh, you guys, I'm not sweating right now, but I'm still losing water that I am unaware of. Okay, so insensible water loss. Your skin is not perfect at keeping you from losing water, and you're losing water through your lungs. And so those are your insensible water losses. You're not aware of them happening. So sorry about that, friends. Oh, well, that's not going to change, is it? Okay, which is the most potent stimulus for thirst? That, because remember, your little cells in your hypothalamus are going to crenate. That is the most potent stimulus for thirst. Yeah, your mouth is dry, but you're going to take one sip of water and then your mouth isn't going to be dry anymore, but you still need to take in water. So this one is the most potent stimulus for thirst. Okay. You were thirsty and drank a glass. Oh, there we go. Only to have your thirst return an hour later. Why? Okay, yeah, because you drank and it stretched and it stopped and then they didn't do it anymore. All right. Okay, when ADH levels are low, the kidney produces what kind of urine? Dilute, absolutely. Yeah, you had it concentrated, but remember ADH concentrates your urine. So if my ADH is low, then I'm going to get a dilute urine. Okay. Runners drinking cups of plain water can become disoriented and collapse. Why? Yes. Okay, because the cell fluids are diluted. Good job. Okay, which best summarizes the relationship between sodium contents of fluids and water movement? Perfect. I like it. All right. Which two hormones are essential for reabsorption of water at the collecting duct? I think we have beat this to death over a semester, ever since the beginning of the semester. So I hope we get 100% on this question and 100% right. Oh, friends, hormones. Okay. Renin is just on angiotensinogen. Aldosterone is on sodium reabsorption, okay, which you need to reabsorb water. But remember, you need aldosterone and ADH for the aquaporins. Aquaporins are not hormones, friends, okay? And again, ADH and renin don't go together. Aldosterone and ADH, don't ever forget that, because that's all part of the renin, angiotensin, aldosterone system. And then ADH is also part of that as well. All right. No, we didn't change anything. Okay. What contains the largest percentage of water in the body? Okay, remember 62, I just said that in the last question, a couple questions ago. 62% of our fluid is inside of our cells, 30% in interstitial fluid, 8% in plasma. Okay, so 2/3 of our fluid is intracellular. Don't forget that. All right. Which of the following is the initial means of regulating hydrogen ions? So the first thing, if I got too many hydrogen ions, what am I going to do? Chemical buffers, absolutely. Good job. Which of the following buffer systems is the major extracellular fluid buffer system? Okay, good. Bicarbonate buffers. All right. How does the pH of intercellular fluid change when the pH, that should be intracellular, but no, so this would be like interstitial fluid. That's a really bad way I worded that, but we just went over it in our things, in our PhysioX. Okay, so increased metabolic rate, what does our pH do? Decreases. Okay, so think about it. Okay, so go back to cellular respiration. That's our metabolic rate. And so we're making more CO2. So as CO2 rises, pH falls. So always remember that relationship. CO2 rises, pH falls. All right. Okay, if the blood pH falls, what change would you expect from the respiratory system? Yes, respiration would increase. We would hyperventilate. Good job. All right, main force driving filtration in the glomerulus is? Yep, glomerular hydrostatic pressure. Look at you guys, woo, 100%. Okay. Pathologies with the lack of blood proteins or protein in the urine can all cause edema by. So anytime I don't have enough blood protein or protein in my urine, why do I have swelling? Okay, yep, no colloid osmotic pressure. No, remember that proteins don't have anything to do with hydrostatic pressure. That's all blood volume. Okay, but I would push out the fluid, but I couldn't draw it back in because I don't have albumin, or my albumin leaked into my interstitial fluid or my urine. Okay. Which of the following would lead to metabolic acidosis? Okay, so diuretic use. Wait a minute. It's diuretics. That's urination. Oh no, that's metabolic alkalosis. Starvation, yep. Vomiting, nope, that's metabolic alkalosis. Constipation, that's metabolic alkalosis. So all of these, we're losing hydrogen ions, whoops, in this, or retaining bicarbs, but starvation is that we're producing hydrogen ions, well, ketone bodies. So that's just off of that table that you had to know with ABG. So make sure you review those pathologies for your bonus questions, your bonus points on the ABGs on the test this week. Okay, what portion is most actively involved in reabsorption of all portion of the tubule? Glucose and amino acids. Proximal convoluted tubule. Yep. Remember, all glucose and amino acids are absorbed by the end of the proximal convoluted tubule. And Macy's climbed back on top again. The mechanoreceptors that detect blood pressure in the afferent arteriole are the okay, the macula densa are chemoreceptors. Okay, so remember always that, chemoreceptors. And they're clear over at the distal convoluted tubule. Juxtaglomerular cells are the ones that are right surrounding the afferent arteriole, right at the beginning. Okay, stretch receptors, pressure receptors. Okay, function of angiotensin II is to okay, so let's think about angiotensin II. Does it decrease production of aldosterone? No, so it can be all the above, right? Because remember, it's the RAS, renin, angiotensin, aldosterone system, which is going to cause an increase in water reabsorption, and angiotensin II is going to obviously constrict the arterioles and increase blood pressure. So no, it's not all of the above. The only one that it's going to do is the gold one. The other ones are opposite to what it does. Okay, what is normal GFR? Okay, this is cardiac output, you guys, for those that you picked that one. So that's a thing. It's a thing. It's just not GFR. So GFR is 125 milliliters per minute. Okay, which is not a mechanism for maintaining GFR during a drop in systemic blood pressure? Okay, so they are all, so tubuloglomerular feedback is I take a look at what the concentration is in my urine and of the sodium chloride, and then I can adjust and I adjust with renin-angiotensin, and I adjust with myogenic, because these are the intrinsic mechanisms, and then this is the extrinsic. So we would use all of them during a drop in blood pressure. So that was the I'm trying to think what color it was on the little chart. The yellow, was it? I don't know. Anyway, it's one of those on the chart. Okay, what would happen if capsular hydrostatic pressure were increased above normal? Yes, because it opposes filtration, so it would cause filtration to decrease. Because remember, it's the pushback. So it's, remember, you have hydrostatic pressure of the glomerulus minus capsular hydrostatic pressure and minus glomerular osmotic pressure. Okay, which causes an increase in the rate of glomerular filtration? Yay! Good job. Because if I increased osmotic pressure, that'd be pushback. If I increase capsular, that'd be pushback. If I decrease, that wouldn't be enough. My GFR would go down. So yep, increasing hydrostatic pressure. Excellent. Which structure best achieves the main function of the urine system? Oh, well, we already did this. Urinary system, the production of urine. Perfect. Nephron. Good job. All right, the kidneys are stimulated to produce renin. Excellent. Good job. pH doesn't have anything to do with blood pressure. So renin is produced during times of low blood pressure. So if I have low systemic pressure, then I'm good to go. Oh, I didn't read the filtration membrane consists of. Okay, so remember, we want the epithelium of the capillary wall, basement membrane, and the epithelium of the capsule, of the glomerular capsule. Okay, so all three of those are the filtration membrane. Oops. In response to increased levels of aldosterone, the kidneys produce. Okay, so if I have aldosterone, what will be the effect? Yes, because we reabsorb sodium, and then what happens when we reabsorb sodium? We decrease our volume of urine. Good. Okay. Nephron cells that respond to the concentration of filtrate are. Yep, macula densa cells. Good job. Juxtaglomerular cells respond to the stretch, blood pressure. Macula densa is this thing. What is it? What is this? Concentration. Hello. All right. One of the roles of the countercurrent mechanism in the nephron is okay, produce a dilute urine. That is exactly right. So by creating that osmotic gradient, then the water comes out, the sodium comes out, and the water stays in on the other side, and then we produce the dilute urine. Okay, the ability to concentrate urine is dependent on the functions of what? So whose job? Or who can do that? Good job. It is the loop of Henle. Okay, so basic functional unit of the kidney is the it is the nephron. Good job. Because the nephron is what does the work. Okay, blood supply to the nephron is the, oh, well, we did this already back in the beginning. Oh, no, we did glomerulus. But what supplies it to the glomerulus? Yes, the afferent arteriole. Perfect. The most important factor affecting glomerular filtration rate is, we already did this, but we're doing it again in a different way. Yep, hydrostatic pressure. Still blood hydrostatic pressure, not capsular, because that's the pushback. This is pushing filtration out. It's always got to be higher, and then we get filtration. Which substance would not be expected in urine normally? Yes, protein. Sweet. Which urine formation process results in acid elimination? So which one is going to make you lose acid? Yep, secretion. Good job. All right, we're getting done. If the efferent arteriole constricts and the afferent arteriole remains unchanged, the GFR will. Or the GFR, something. Yep, it's going to increase because you're going to get it backed up, and so more blood will stay within the glomerulus. Was the main substance leaving the descending limb of the loop of Henle? Yes, water leaves the descending limb. Good job. Okay, presence of glucose and ketone bodies in the urine can indicate what is wrong with you. correct. Yay. So proud of you. GFR is regulated mostly by. So what really controls GFR? I mean, make sure that it stays where it's supposed to be. That's what that meant. Yep, autoregulation, myogenic mechanism, and we kick in the hormones only when it's super low, when we go systemic, and there isn't really well, sympathetic stimulation, I guess, but that's these two. So the gold and the blue are ones when we absolutely have to kick in. Otherwise, we're going to do autoregulation all the time. And 3 more. Which of the following statements about the urinary system is incorrect? Okay, gluconeogenesis happens in your liver. So yes, it is a long-term regulation of the systemic blood pressure. It does produce erythropoietin. It does maintain fluid and electrolyte balance. It does not do gluconeogenesis. That's your liver making glucose from other substances, fatty acids, proteins. Okay, 2 more. Which of the following statements about juxtaglomerular cells is incorrect? Yep, this would be the macula densa. Otherwise, the other ones are all right. And the last question, yay, is all the following are layers of the filtration membrane and the glomerular membrane, except okay, so yep, there's not renal capsule because it's visceral membrane, fenestrated endothelium, and basement membrane. And there it is. Woo! Yay! Woohoo! Okay, so that's it. So good luck with your test this week. It's all in there and ready to go, but it's just open first thing in the morning, and you have till Saturday at 11:59 to get it done, and then the lab practical will open tomorrow as well. And there it is. So tomorrow's lecture will start male reproductive system. So have your reproductive system notes ready to go. And there it is. Let's get out of that. Okay, leave. All right. So have a great everything rest of the week, but I'll still see you tomorrow. So, okay, I don't have anything else. We've been here long enough. Okay, take care. Have a good day. Bye.